Okay, so here's part 2 of our genetics lesson. By now, you should have memorized all of the genetics vocab that we went through in the earlier lesson and also know how to apply it, because here's where the application of those genetic vocab words are going to come really handy, because now we're going to apply those words in these word problems called Punnett square crosses. Now, the best way to go about the Punnett square cross is to always give you an example. So, the example that we're working with is called the monohybrid cross. You're just looking at one single trait. Now, Gregor Mendel actually formulated these monohybrid crosses, and so we're going to take a look at his work and then later apply these same rules to different kinds of problems. So in Mendel's one trait cross, the trait or the gene that he was looking at was the actual seed color of the plant. Now, for every gene, there's always going to be a dominant allele and a recessive allele. In this case, the dominant allele, which you know should be dominant because it is a capital letter here, okay, there's the capital letter, so you know it's dominant, is actually yellow. So yellow is the dominant color. Lowercase g is the recessive. A lowercase letter is always representative of the recessive allele. So in this case, it's going to stand for green. Now, there's other traits that he looked at, but again, we're focusing on just one trait, and this is the seed color. So his first Punnett square problem is called the parental generation, also known as the P generation. In the P generation, the rules are that both parents must be homozygous. Parent number 1 is homozygous dominant, so parent number 1's genotype is big G, big G. Parent number 2 has to be homozygous recessive, so their genotype is little g, little g. Again, remember that genotype are the letter combinations. These are all genotypes. We cannot see this when we look at a person. Okay, so don't expect to look at someone and see letters. No, we just do this on paper so we can map out the problem. Now, phenotype is the physical description of what the color of the seed is. So if parent number 1's genotype is big G, parent number 1's phenotype should be the dominant color, which in this case is yellow. Parent number 2's phenotype, because the genotype is little g, should be green. Okay, so again, notice the difference between geno and pheno. Geno are the two letters. Pheno is what you are actually using to describe what the seed color is. So anytime you're working with a parental generation Punnett square, these are the rules that you have to follow. If I were to ask you to set up a P generation problem, you have to follow these rules. These rules will never change. Okay, so moving on. Example is the Punnett square cross. In order to use a Punnett square, the parents' genes go on the outsides, like so, and it doesn't matter where you put mom or dad's genes, just as long as they go on the outside. But the chances of the offspring's genotypes belong inside the boxes, where you just simply connect the letters that are on the outer edges and always put the capital letter first. So like so. Okay, so that belongs in the first box. That is why that's big G, little g. This guy goes right here. Okay. This guy and this guy. And this guy and this guy. Okay? It may look confusing, but it's super easy once you get the hang of it. Again, remember, parents' genotypes always go on the outside. So this is parent number 1, and this is parent number 2. Okay, moving on. So, the P generation, when you're reporting back probabilities, you'll notice that inside all 4 boxes, all the genotypes are the same. Big G, little g, they're all heterozygous, and their phenotypes are 100% yellow. So, probability is the likelihood that an event will happen. It's always either represented in a fraction form or a percent form. The fraction form tells us the number of favorable outcomes, which is the numerator, over the total number of events, the denominator. The denominator in this case will always be 4 because there are 4 Punnett squares inside the box there. So for the P generation, you can say either 4 out of 4 or 100% have the genotype probability big G, little g, and 4 out of 4 or 100% have the phenotype probability of being yellow. So the next problem is the F1. The F1 generation does not require a Punnett square. You are simply just reporting back the probability that you got from the P generation. So again, it's a simple repeat. So for the F1 generation, which are the children from the P generation, the probabilities are 100% or 4 out of 4, big G, little g as the genotype, 100% or 4 out of 4 yellow as the phenotype. Okay? So again, anytime I ask you to set up an F1 generation problem, you are simply just going to report back the probabilities from the P generation. The last and final problem is F2. This is the 3rd generation. For every F2 generation problem, you are crossing 2 of the F1 children. So that means that both parents must be heterozygous. So again, the parents' genotypes go on the outside, like so, and then you simply fill in the inside 4 boxes according to the letters that appear on the outer edges. So again, this guy and this guy, this guy and this guy, this guy and this guy, and this guy and this guy. Remember, capital letter always goes first. Next step is to report back the probabilities of what shows up in each of the 4 boxes. When you report back probabilities, remember you're reporting back both genotype probabilities and also phenotype probabilities. So if you look at the inside boxes, this is what you should report back. There are 3 possible genotypes that show up. Big G shows up 1 out of 4 or 25%. Big G, little g shows up 2 out of 4 or 50%. And little g shows up 1 out of 4 or 25%. Again, for every single F2 generation probabilities, these are the same exact results that you should get because you should have set them up in the same way because you follow the same rules. The phenotypes, yellow shows up 3 out of 4 times, which is 75%, and green, 1 out of 4, 25%. So hopefully you got these results and now you can apply them to different scenarios. Make sure that you look back to this video if you need to. But again, the rules for P generation, F2, and F1 will never ever change. They are outlined for you in this video, so please make sure that you always look back to it.